Hello friends, in this video we are going to discuss about DNA polymerase in prokaryotes. It was first purified and characterized by Arthur Kornberg in E. coli. What are DNA polymerase? These are group of enzymes that catalyze the synthesis of DNA strand on template strand during DNA replication. So, these catalyze the synthesis of DNA molecules from nucleoside triphosphates, and these nucleoside triphosphates are molecular precursors of DNA. Now the functions of DNA polymerase. First, DNA replication. It is the main function. DNA polymerase duplicate or replicate DNA content of cell during cell division. Thus, genetic information is maintained and transferred from one generation to another. Second is repair. It maintains integrity of genome. How does DNA polymerase work? Main function of DNA polymerase is DNA replication. Existing DNA strand is read and two new strands are created, same as existing one, so that each daughter cell during the cell division receives similar DNA molecules. Chemical reaction that is catalyzed by DNA polymerase during replication is DNA N. N indicates N number of deoxynucleotide triphosphate plus 1 deoxynucleotide triphosphate. Now DNA is increased by 1 deoxynucleotide triphosphate represented as DNA N plus 1 plus inorganic phosphate. Direction of replication is 5' to 3' always. Why? Because DNA polymerase can add the nucleotide at the 3' end only, and one nucleotide is added at a time. So the directionality of the daughter strand, which is 5' to 3' is opposite to the direction of the template strand, which is 3' to 5' along which DNA polymerase moves. As a result, resultant double-stranded DNA is composed of antiparallel strands. So these two are the parental strand or the template strand. DNA polymerase moves in 3' to 5' direction, here and also here, and the new strand is synthesized in the 5' to 3' direction, this strand and also this strand, as a result of which the new DNA molecule that is obtained has antiparallel strand as the parental DNA molecule. One has 5' to 3' in this direction and the other has 5' to 3' in this direction. DNA polymerase can add nucleotides at 3' OH group but cannot initiate new strand, hence primer is needed. What are primer? These are segment of DNA, RNA or both. Generally it is RNA. Now the processivity of DNA polymerase. It is average nucleotides added by a DNA polymerase per association with the template strand. So, DNA polymerase associates with the template strand for the synthesis of new strand, and these add nucleotides one by one in order to synthesize new strand. So, number of nucleotides or the average nucleotides that DNA polymerase adds in one association is known as DNA polymerase processivity or processivity of DNA polymerase. Replication is highly accurate process, and if there is change in single nucleotide, it leads to mutation. There are two mechanisms by which DNA polymerases make sure that there are no errors. First is the geometry of active site. Only correct nucleotide base pair fit. So the geometry of the active site of DNA polymerase is such that it allows only the correct nucleotide base pair to fit. But still, incorrect nucleotide is added, but it is rare. It is added after 10 power 4 to 10 power 5 nucleotides. 3'5' exonuclease activity of the DNA polymerase, which is known as the proofreading, helps in the removal of any error if it is incorporated in the DNA. So in this proofreading activity, when incorrect base pair is recognized, DNA polymerase move backwards by one base pair. Incorrect base is excised. This is followed by reinsertion of correct base. This is known as proofreading. In this way, integrity of the original DNA strand is preserved and it is passed on to daughter cells. But still errors arise in the DNA, and this is due to replication errors and errors due to DNA damage. DNA is damaged by various factors. Among them, one of the most important is by the UV radiation. It is important to maintain the fidelity of the DNA during the replication, or the fidelity is very important in the DNA replication, because the mismatches in the DNA base pairing result in dysfunctional protein and can lead to cancer. So it is very important to detect the error that is incorporated in the DNA strand and how this error is detected. The shape and the interactions that accommodate Watson and Crick pair contribute in the detection of error. We have discussed that DNA has double helical structure, which was proposed by Watson and Crick, where adenine always pairs with thymine and guanine always pairs with cytosine. But if any wrong base is incorporated, then it can be detected and the error can be removed. So how it is detected and then the correction of the error is done at a mismatch, that is, if there is mismatch base pair, then there occurs loss of interaction between the base pair, and this loss of interaction triggers shift in balance from polymerase to exonuclease domain for the binding of the template primer. Now, the exonuclease domain binds to the template primer in order to correct the error. DNA polymerase has polymerase and exonuclease domain, which we will discuss in detail in the next video, where we will see types of DNA polymerase, their structure and function. So if we see this figure, this is the template strand and this is the new strand which is synthesized by DNA polymerase. If we see here, template has cytosine strand and cytosine always pairs with guanine, but here adenine is added. So here incorrect base is added and there is mismatch, as a result of which these two base pairs do not interact with each other. This results in the shift from the polymerase domain to the exonuclease domain. Now it functions as 3'5' exonuclease. It moves backward by one nucleotide. Incorrect base, that is adenine, is removed and correct base guanine is added, and then again there is shift from exonuclease domain to polymerase domain and replication continues. This is known as proofreading. So what we can say about proofreading, that when wrong nucleotide is incorporated, it causes retardation in the DNA polymerization. And due to this delay, DNA polymerase is switched from polymerase site to exonuclease site, then the error is corrected and then DNA replication again continues. DNA polymerases are also involved in post-replication repair of DNA, which we will discuss in detail in separate video on repair mechanism of DNA. Now fourth is translesion synthesis. To avoid the harmful consequences of a stalled replication fork, certain specialized polymerases, example polymerase 4 and 5, traverse the damage. When there is damage or lesion in the template strand, then DNA polymerase 3 stops, extending the new strand. as a result of which replication fork is stalled. In that case, certain specialized polymerases, example polymerase 4 and 5, which are also known as SOS polymerase, travel past the damage and continues the extension of new strand. And this is called translesion DNA synthesis or TLS. This provides the cell additional time to repair the damage, and once the damage is repaired, replicase, in this case DNA polymerase 3, returns to complete genome duplication. Let us understand it in this way. When lesion is encountered in the DNA template during the replication fork stalls or DNA polymerase 3 stop extending the new strand or replication of the DNA. DNA polymerase 3 then dissociates from the template strand and it is replaced by translesion DNA polymerase, example DNA polymerase 4 and DNA polymerase 5. These are known as SOS DNA polymerase. These extends the DNA synthesis across lesion or damage, that is, these traverse the damage or travel past the damage and continues to extend the DNA synthesis. In this way, cells get additional time to repair the damage. And once the damage is repaired, replicase, that is DNA polymerase 3, returns to complete duplication of DNA. So this is all for today's video. In the next video, we will discuss about types of DNA polymerase in prokaryotes. We will discuss about their structure and function in detail. So stay tuned. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.